Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study from the Journal of Veterinary Internal Medicine, published in 2024, that explores the potential of certain substances in the blood to predict the outcome for cats suffering from feline panleukopenia. This research, conducted by Kerem Emre Yaner and colleagues at Ataturk University in Turkey, investigates the roles of copper, zinc, the ratio of copper to zinc, cobalamin, and serum amyloid as early indicators of whether a cat with a serious viral disease is likely to survive. Feline Panleukopenia virus, often referred to as FPV, is a highly contagious pathogen that poses a significant threat to both domestic and wild feline populations. The virus targets rapidly dividing cells, with a particular impact on the cells lining the intestinal system. This viral activity leads to inflammation within the intestinal lining, increasing the permeability of the intestinal wall and hindering the absorption of essential nutrients. The study highlights the importance of cobalamin, also known as vitamin B12, which is crucial for several key enzyme systems involved in the synthesis of nucleic acids and the production of blood cells. Cobalamin is absorbed from food through a complex process involving receptors in the intestines. In cats, the pancreas is the exclusive site for the production of intrinsic factor, a protein necessary for cobalamin absorption in the final section of the small intestine, called the ileum. Previous research has indicated that reduced levels of cobalamin can be associated with changes in the bacterial populations within the small intestine or with gastrointestinal diseases, suggesting it could be a valuable marker for assessing the extent of intestinal damage caused by FPV. Copper and zinc are trace elements absorbed by the intestinal epithelial cells in cats and serve as essential components or cofactors for numerous proteins vital for various bodily functions. Research in humans has linked these trace elements and their ratio to conditions like gastrointestinal inflammation, impaired immune function, increased oxidative stress, and malnutrition. Recent studies in dogs have also suggested that trace elements can be reliable indicators of the likely progression of inflammation. Therefore, investigating the prognostic value of serum concentrations of these trace elements in cats with FPV appear to be a promising area for enhancing routine veterinary practice. Acute phase proteins, or APs, are substances in the blood whose concentrations change in response to inflammation. Their use in clinical settings has been increasing, with recent studies emphasizing their role not only in diagnosing diseases, but also in predicting how a disease might progress and monitoring the effectiveness of treatment. Serum amyloid A, or SAA, has emerged as a significant AP in cats, and while its use as a diagnostic marker is becoming well established, its potential for predicting the outcome of diseases in routine clinical practice is still being explored. This study aimed to evaluate the potential of copper, zinc, the copper to zinc ratio, serum amyloid A, and cobalamin as prognostic biomarkers in cats with FPV, specifically during the early stages of the infection. The researchers also sought to determine how effective these biomarkers were at predicting the final outcome of FPV using important measures such as positive predictive value, sensitivity, cutoff values, and specificity. Measuring the concentrations of these substances in cats infected with FPV could help veterinarians better monitor the likely course of the disease during a cat's stay in the hospital. To conduct their research, the team performed a prospective observational study involving client-owned cats that presented with diarrhea at the small animal clinic of the Faculty of Veterinary Medicine at Ataturk University. The study included cats diagnosed with FPV based on clinical signs like lethargy, vomiting, hemorrhagic diarrhea, anorexia, and dehydration, along with a lack of vaccination against the virus. The initial screening for FPV involved a rapid antigen test, and the diagnosis was further confirmed using polymerase chain reaction, or PCR, to detect the virus's genetic material in fecal samples. Cats with other concurrent diseases were excluded from the study. A control group of healthy cats, matched for age, was also included for comparison. Before any treatment was administered, blood samples were collected from all the cats to measure the concentrations of copper, zinc, serum amyloid A, and cobalamin. Standard laboratory methods were used for these measurements, including spectrophotometry for copper and zinc and chemiluminescent immunoassay for serum amyloid A and cobalamin. The ratio of copper to zinc was then calculated. The cats diagnosed with FPV received standard veterinary care, including intravenous fluids, antibiotics, antiemetics, and immunostimulants. Of the 19 cats with FPV, 10 survived and 9 did not, with all non-survivors dying due to the progression of the disease without being euthanized. The statistical analysis of the data revealed significant differences in the concentrations of the measured substances between the healthy control group, the FPV survivors, and the FPV non-survivors. The researchers used receiver operating characteristic curve analysis to determine prognostic cutoff values for each biomarker, along with their sensitivity, specificity, and area under the curve, which indicates the overall accuracy of the test. The positive predictive value for survival was also calculated for each biomarker on the day the cats were admitted to the hospital. The results of the study showed that on admission, cats that survived FPV had significantly higher serum copper and serum amyloid A concentrations and a higher copper to zinc ratio, but significantly lower serum zinc and cobalamin concentrations compared to the healthy control cats. Furthermore, the cats that did not survive FPV had even higher serum copper and serum amyloid A concentrations and copper to zinc ratios, and even lower cobalamin concentrations than both the survivors and the control group. Specifically, the median copper concentration was significantly higher in both the survivor group and the non-survivor group compared to the control group, and it was also significantly higher in the non-survivors compared to the survivors. A cutoff value for copper greater than 120.3 micrograms per deciliter on admission had a 90% positive predictive value for survival. The area under the curve for copper as a predictor of survival was 0.93, indicating excellent predictive ability, with a sensitivity of 100% and a specificity of 80%. Conversely, the concentration of zinc was significantly lower in both the FPV survivor and non-survivor groups compared to the control group. However, there was no significant difference in zinc concentrations between the cats that survived FPV and those that did not. The ratio of copper to zinc was significantly higher in both the survivor and non-survivor FPV groups compared to the control group, and it was also significantly higher in the non-survivors compared to the survivors. A copper to zinc ratio greater than 1.34 on admission had a 90% positive predictive value for survival. Similar to copper, the copper to zinc ratio was an excellent predictor of survival, with an area under the curve of 0.95, a sensitivity of 100%, and a specificity of 80%. Serum amyloid A concentrations were significantly increased in both the FPV survivor and non-survivor groups compared to the control group, with significantly higher levels observed in the non-survivors compared to the survivors. A cutoff serum amyloid A concentration greater than 0.85 milligrams per liter on admission resulted in a 90% positive predictive value for survival. Serum amyloid A demonstrated excellent diagnostic performance, with an area under the curve of 0.98, a sensitivity of 100%, and a specificity of 80% in predicting survival. Cobalamin concentrations were significantly lower in both the FPV survivor and non-survivor groups compared to the control group, and notably, the non-survivors had significantly lower cobalamin levels than the survivors. A cutoff cobalamin concentration of 430.4 picograms per milliliter or less on admission achieved a 90% positive predictive value for survival. Cobalamin also showed excellent diagnostic performance, with an area under the curve of 0.98, a sensitivity of 100%, and a specificity of 80% in predicting survival. The researchers discussed their findings in the context of previous knowledge about these biomarkers and their roles in disease processes. The marked increase in copper concentrations as the prognosis of FPV worsened aligns with studies in other animals and humans that have observed increased copper levels following inflammation-induced intestinal damage. Intestinal damage caused by parvovirus can disrupt the normal absorption of copper, potentially leading to its increased leakage into the bloodstream. The significant decrease in serum zinc concentrations in cats with FPV, particularly in non-surviving cats, is consistent with human studies linking reduced zinc levels to damage of the intestinal mucosal barrier in various intestinal diseases. This decrease could be due to malabsorption caused by intestinal damage or loss of function of proteins responsible for zinc uptake. Interestingly, the researchers also suggested that the increase in copper concentrations due to intestinal inflammation might contribute to zinc loss, as copper and zinc absorption are known to be antagonistic. The significantly higher copper to zinc ratio in FPV non-survivors compared to survivors and the control group may also be related to the severity of inflammation and the antagonistic relationship between copper and zinc absorption. The substantial increase in serum amyloid A concentrations in both FPV groups, with higher levels in non-survivors, is consistent with its role as a major acute phase protein in cats and the well-established involvement of APS in inflammation. This finding supports the validity of serum amyloid A as a valuable indicator of inflammation and prognosis in various feline diseases. The significantly lower cobalamin concentrations in FPV-affected cats, especially in non-survivors, underscore its potential as a prognostic indicator. While the exact mechanisms behind hypocobalaminemia in cats with FPV remain to be fully elucidated, it is known that gastrointestinal disease can lead to decreased cobalamin levels due to intestinal injury, changes in gut bacteria, pancreatic dysfunction, and increased utilization of cobalamin during inflammation. The study's findings suggest that cobalamin exhibits a negative acute phase protein-like response in cats with FPV, making it an excellent marker for assessing prognosis. The researchers acknowledged that the study had a relatively small sample size, although the number of cats meeting the study criteria represented a substantial proportion of the hospital's admissions during the study period. They also noted that economic constraints limited a more comprehensive analysis of the roles of these promising prognostic markers in the development of the disease. Future research with larger sample sizes is warranted to further evaluate the effectiveness and accuracy of the proposed cutoff values for therapeutic use. In conclusion, this study highlights that cats that do not survive feline panleukopenia have significantly higher concentrations of copper and serum amyloid A and significantly lower concentrations of zinc and cobalamin compared to cats that do survive. Furthermore, serum amyloid A and cobalamin demonstrated remarkable prognostic capabilities. Copper also showed good prognostic potential, and the assessment of zinc concentrations was promising for predicting survival. These biomarkers offer a potentially cost-effective way to predict the likely outcome for cats with FPV at the time of hospital admission. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.